Welcome to the second lecture on Let's Crack NEET UG English. And in the previous one, we talked about the basic information on cell cycle. Now, in this particular lecture, we'll be taking everything in detail. That means we'll talk about the phases of cell cycle, what all changes take place during cell cycle, and also the checkpoints which we talked earlier. The checkpoints we will discuss in detail and how they are beneficial. And then maybe we'll have a couple of practice questions to understand whether you have understood this concept clearly or not. And as I told you, we are on NEET, Let's Crack NEET UG English. I'm your biology teacher, Dr. Neela Bakore, and I'll be taking all these lectures for you. We have started with cell cycle. We'll follow this up with cell divisions, mitosis, meiosis, and all those stages. The platform which we are using for these lectures is Unacademy platform, and this is the largest learning platform in India. What you need to do to take benefit of this platform is very simple. You have to just install the Unacademy Learning app. Once you install this app, you will get access to daily live classes by experienced educators. We have planned the structure in a nice manner so that your complete syllabus is covered. There will be tests conducted on daily or regular basis, live tests and quizzes, and you will also have unlimited access. The live classes are conducted by various educators who have experience in the same field, that is, tutoring or mentoring the students who are preparing for NEET UG. This is just to give you an idea of the fee structure. And if you observe this tabular thing in a very simple manner, you can easily understand that you get benefited financially if you pick the plan which is for a longer duration. So say if you go for a 12-month plan, it is less because it is a very simple math. 1 month is 5,400, you multiply it with 12, that is for 12 months. You get some figure. But if you look at this figure here, it is definitely less because you are getting a discount. And if you want to avail 10% discount, you have to use my code NEELA when you subscribe. So you would get that benefit. And you can even go for a longer duration. So you can choose the plan according to your convenience. Let us come back to the topic. The cell cycle is what we were talking about. Now, I have given you a brief introduction of a cell cycle in the previous lecture. And we have also talked of some basic things which happen. Here, we will take all those changes in detail in every phase. So, from here, that is once the cytokinesis is over, complete change which is taking place up till this point is the first phase, that is interphase. This is interphase. And in interphase, we said there are 3 subphases, G1, S and G. Now we are trying to see what exactly happens in G1 phase. So let us see what happens here. G1 is a preparatory phase for S. The cell prepares itself for the next phase, that is S. So we need to write something in S phase first. And in this phase, there will be DNA replication. DNA is going to copy itself. So from one, we will have 2 DNAs. For this, there are enzymes required. And we know that the enzymes are proteins. So what is going to happen here in G1 is protein synthesis. Which kind of proteins are we talking about? these proteins are mainly enzymes. If you are studying cell cycle for the first time, that means if you are in class 11, this is first time when you are talking about it. So probably you are not aware of the names of the enzymes. But those who have completed the 12th and they have done this particular part of DNA replication, they know that there are 10 enzymes which are required. Helicase, topoisomerase, DNA polymerase 1, DNA polymerase 3, many enzymes. So all those enzymes must be prepared. So enzymes required for DNA replication. These enzymes are synthesized here. This is the main protein. Now, do we know that how proteins are synthesized? In our body, the genetic material, that is DNA, has all the information. Whatever happens in our body, that information is on DNA. And DNA is inside the nucleus. So, if a protein is to be synthesized, for which the information is on DNA, and protein synthesizing structure, that is ribosome, is in the cytoplasm. DNA cannot come out of nucleus and ribosomes cannot go inside the nucleus. So how is this information from DNA brought to the site where protein is to be synthesized? There has to be some molecule which is going to bring this message, and that molecule is mRNA. So this is actually known as central dogma. So from DNA, the information is brought by messenger RNA, and this messenger RNA attaches to ribosome and where protein synthesis takes place. So protein is formed, and this whole thing is known as central dogma. This is important. Without this central dogma, life cannot exist. So if we want to synthesize enzymes which are proteins, first of all, RNA should be made. Like here we've written mRNA. With ribosome there is rRNA. Then there is one more RNA which is called tRNA, which is going to bring the amino acids. So before protein synthesis, anywhere, it is going to be RNA synthesis first. So here RNA synthesis is going to increase and then protein is synthesized. So this protein is basically the enzyme that we're talking about, is synthesized in this manner. First from DNA to RNA, then RNA to protein. This preparation is taking place in G1. Energy is also required. There are so many changes, so many things taking place inside the cell. So ATP, which is the energy currency of the cell. So ATP is also synthesized and is accumulated inside the cell. Now, we want DNA to replicate. When DNA replicates, that means on one original strand, one more copy strand will be formed. This copy strand will require the raw material. So how are you going to make new DNA without raw material? And what is that raw material? The raw material is called nucleotide. So here, nucleotides, which is the raw material, is also synthesized. So now, is the cell ready to enter S phase? What is required for S phase? DNA is to be replicated there. So are the enzymes ready? Yes, they are. Is sufficient ATP or energy accumulated? Yes, it is. Is the raw material ready? Yes. So now the cell can enter S phase. In S phase, everything is, all the preparation is already done in G1. And in S phase, all that is used for DNA to replicate. So here DNA replication will take place. Along with that, there are two more things which happen in S phase. One is centriole splits. Centriole is going to help in assembly of spindle fibers when the cell is going to divide. So centriole also splits in this S phase, and histone proteins are synthesized. Where is this histone protein required? Are you able to recall the first lecture? We talked about the structure of chromosome. Inside the chromosome there were chromonemata threads. And we also said that the DNA which is present in one cell or the nucleus of one cell, if you combine, if you stitch all that DNA end to end, it is going to be 2.2 meters long. So how do we package such a long DNA in a microscopic cell? This is possible because of coiling and supercoiling of our DNA. And where is this DNA coiled? It is coiled around histone protein. So if you are making new DNA, you have to make arrangement so that that DNA can be accommodated in that nucleus, and for which this histone protein is synthesized. So whatever was done in G1 has been used up here. Enzymes, ATP, raw material, everything has been done here. So S phase, mainly three things: DNA replicates, centriole splits, and histone proteins are synthesized so that the newly formed DNA can coil around it. Now the cell comes into G2. In G2, the cell is going to prepare itself for division, entrace. So what is required for division? Let us see. What is required for division? The cell is going to divide. Energy will be required. Proteins will be required. Which proteins? Now here there are two categories of proteins. So these are the changes which take place in G1, and here we will write down the G2 changes. Okay. So what is going to happen in G2? Protein synthesis. And tell me, are the proteins going to be synthesized like that only? Before that, RNA has to be synthesized. Wherever protein synthesis takes place, RNA has to be there. So here, RNA synthesis also increases. All three types of RNA, mRNA, rRNA, tRNA, all of them. And then the proteins. The proteins are of two categories. One category is of enzymes. Which type of enzymes do we need? Why are these enzymes required? The enzymes are of two types. One, lytic enzymes. Lysis is to break down. See, the cell has to divide, but before the cell divides, the nucleus is going to divide, the genetic material is going to divide. So, there has to be enough space for the chromosomes to move. And if everything happens inside that tiny nucleus, then the space is very less. So what happens is all the membrane bound structures, all the organelles, they lose their membranes. Dissociation of the membrane will take place. And that will be done by these lytic enzymes. So in the beginning, when the chromosomes have to move, lytic enzymes will dissolve or dissociate that membrane. But later on, all those organelles must assemble. So for assembly, that means you have to reassemble that membrane. So for that, we need one more category of enzyme, and those are the synthetic enzymes. So one category of protein is going to be enzyme, two categories of enzymes, lytic and synthetic. Then one more protein is required, which is tubulin protein. Tubulin protein is required to make spindle fibers. So spindle fibers are made up of tubulin proteins. And when I say that the chromosomes have to be moved, how are they going to move? They are pulled by these spindle fibers. And that spindle fiber is made up of tubulin, so we need that protein also. So what is happening in G2 is preparation for division. So the cell has synthesized protein in this phase, enzymes as well as tubulin. And to move chromosome from the midpoint of a cell to the pole towards one end, almost 30 ATPs are required. So the amount of energy required is also very high. So ATP has to be synthesized. Now, do we think that the cell is ready to enter into M phase? So now the cell enters M phase. In M phase, first the nucleus is going to divide and then the cytoplasm. So this part is karyokinesis, where first phase is prophase, metaphase, anaphase and telophase. In these four phases, the nucleus divides and then the cytoplasm division takes place, that is cytokinesis. After cytokinesis, one parent cell would divide into two daughter cells. And if these two daughter cells do not have to divide, then what is going to happen is they will enter a phase where the cell becomes inactive division-wise, and that is known as G0. So this is G1. This whole part is G1. From here till here is G1. So these two daughter cells have entered G1. They'll grow to their normal size and then they go into G0 or G0 phase. So this G0 phase is known as quiescent phase. Quiescent means inactive, but here we are talking about inactive only in respect of division. The cells which enter G0 phase or G0 phase, they become inactive with respect to division only. They perform specific function. They are very active for all the activities for which that cell has been formed. But it is not going to divide. There is an exception and we will write that exception also. So what happens in G1 is preparation for S. What happens in G2 is preparation for N phase. Right? So we have written down the specific changes which take place during cell cycle in every phase. Now, I always recommend that you draw the diagram and write down everything around that diagram so that when we revise, it becomes very easy. But in our subjective exams, like if we have to appear for 11th class and 12th class exams, which are subjective exams, we have to write down things. So there you have to draw the diagram and then write down the points. So what I have done is, I have also written down these points separately for you. Same thing, what we did along with the diagram, same things are written here. So let us see what is happening in G1. There is increase in protein synthesis, we wrote. Nucleotide, that is the raw material, has accumulated, and because of which, or rather for protein, there was RNA synthesis. And when so many things are getting added into the cell, there is an increase in size of the cell. Now, can we give a name to this G1? Can we call it post-mitotic and pre-synthetic? Let us see this. It is before synthetic phase. It is before synthetic phase, so we call it pre-synthetic. And it is after M phase, so we call it post-mitotic phase. Okay? So, naming of this with respect to the phases which are before that and after that. It's not very important thing, but sometimes such names are also given. But most common is G1 phase. So now we have understood how we are going to write it if it is a subjective name. But when you are learning it, it should be done along with the diagram. S phase, synthetic phase, where DNA is going to be synthesized or replicated. So DNA replication takes place. Histone protein synthesized, centriole splits. Okay. Let us understand this point which we have written here. Each chromosome has two sister chromatin. We have seen the structure of chromosome. Say this is our chromosome in G1 phase. DNA replication has not taken place yet. After DNA replication takes place, what is the change which is seen? So what happens here is, say this is the original DNA of this chromosome. The original DNA. And here is this S phase. So what happens in S phase? The original DNA replicates, and replicates means we get a copy of it. So there is a copy of this. And when the copy is formed at that time, a membrane is formed in this arm. This arm is chromatid. Okay, so there is only one arm on one side, other arm on the other side. What do we see in the chromosome after S phase? If we make this is one arm which is having one DNA copy, and this is the other arm which is having the other DNA copy. So these two arms are called sister chromatids. And are they attached together to the same centromere? So what has happened after S phase? DNA replication. But this DNA has replicated inside the same chromosome. Here, from outside, we would see only one arm. We don't see two arms. But there is a separation inside. So these two parts are called the sister chromatids. So has the number of chromosomes changed? The answer is no. But one chromosome has two sister chromatids. Whereas here, one chromosome is having only one chromatid. So in S phase, DNA replicates, centriole splits, histone protein is formed. But because of this DNA replication in each chromosome, we see two sister chromatids held together at the same centromere. Such type of questions are also asked in competitive exam. That if you have 4 chromosomes in a cell, and those 4 chromosomes have completed S phase, how many chromatids will be visible to you? Or how many chromatids do they have? So we'll have to calculate this way. G2 or gap2, what are the changes which are taking place here? Here the nuclear volume increases. In gap1, the cell size increased. In gap2, the nuclear volume increased. RNA synthesis, protein synthesis, the protein is tubulin, which is required for spindle fiber formation. ATP has to be formed, and the cell is going to divide now. So organelle should also duplicate, so that both the daughter cells should get sufficient number of organelle. And when those daughter cells grow in G1, they'll get more and more organelle made. But initial division, they should have some organelle in them, so that the basic functions can take place. So this is G2. M phase, the cell is going to divide here. So now in M phase, what happens is, first is going to be the karyokinases and then cytokinases. Karyokinases is completed in 4 phases or steps: prophase, metaphase, anaphase and telophase. We will be doing detailed study of these divisions, mitotic divisions and meiotic divisions, in later lectures. And cytokinases is division of cytoplasm. So this is how our cell cycle gets completed. Let us talk about G0, G0 phase. This is very important, and there are many questions which are asked on this. It is known as the quiescent phase, and as I told you, this means the cell is inactive only in terms of division. Otherwise, it is specialized cell and it is going to perform specific function. The cell gets differentiated. So, if I make that cell cycle again for you, just for reference, this is our short M phase. This is our G1, S and G2. So, we say that the cell comes out of the cycle. This is G0. And this process where the cell comes out of the cycle, gets specialized, is known as differentiation. And cells which get differentiated, they remain in G0 and keep performing that specific function. They do not divide after that. That means they never come back into the cycle. But there is an exception, and the exception is parenchyma. So, suppose this is a parenchyma cell. Parenchyma cell which has differentiated. That means it is in G0. It is going to perform its functions. What are the functions of parenchymal cells? They act as temporary storage cells. If they have chloroplast, they will help in performing photosynthesis and so on and so forth. But if a plant requires that this parenchyma cell has to divide, then the cell is going to come back into the cell cycle. The cell enters G1. Here it will prepare itself for S. It gets into S phase, G2, division, N phase. And now parenchyma is going to divide. So, from G1 going out into G0 was termed as differentiation. What should be the reverse movement called? The reverse movement is known as de-differentiation. So if a specialized cell, if a differentiated cell gets back into the cycle, then we say that the cell has de-differentiated. It comes back into the cycle and starts following the same G1, S, G2, M and so on. Okay? So this example we have to remember, parenchyma cell after becoming specialized, after becoming differentiated or after getting differentiated, they can come back into the cycle. This is G0. How long does the cell cycle last? There are three examples which I give you here. Duration varies. It is 20 minutes in bacteria, 8 to 10 hours in intestinal cells, 20 hours in onion root tip cell. So, depending upon the cell, the timing is going to be different. When we talk of bacteria, in our NCERT it is mentioned that it takes 20 minutes for a cell cycle to get completed, or we can say for a cell to divide. So, 20 minutes to divide. And in this, maximum time is taken for DNA to divide. So 18 minutes actually are required for DNA replication. So if a question is asked, like how much time is required in bacteria for replication, our answer is going to be 18 minutes. And if the question is how much time is required for bacteria to divide, our answer is going to be 20 minutes. So 18 minutes are for DNA to divide and then 2 minutes take place for cytokinesis. So total 20 minutes. This is very important. Questions have been asked on this particular point. We were talking about the checkpoints. When a cell divides, there are chances that some things can go wrong. There are so many things taking place. There are so many things which are produced in G1. There is DNA which is getting replicated in S. Then again preparation in G2 and then the cell division. Suppose something goes wrong, then what do we do? If something goes wrong, the new cells, the daughter cells which are formed, they are going to be faulty. They are going to be mutated. And such cells can result into complications like cancer. So there must be some checks that everything is going properly. See, it's a very simple thing. When we go from one state to another state, suppose we are going from Madhya Pradesh to Maharashtra or from Gujarat to some other state, there are some interstate borders. And on these interstate borders, our vehicle is checked. They ask for the papers. Do you have proper papers or not? Do you have license or not? Reason is they want that no illegal thing should happen. No such car should enter their state which has been stolen or which has some other history behind it. So they're just checking that everything is proper. Same thing happens for the cell. And we are talking about the cells of our body. They must be proper and perfect cells. So in our cell cycle, there are 3 checkpoints. And in these 3 checkpoints, all those things which are happening are going to be checked. So let us start from this. This is the first we are going to start from. This is called G1 checkpoint. It is also known as G1S checkpoint. Okay. Is there a question in your head? How are these checkpoints going to work? I'll give you an example of a vehicle entering into a state, interstate border, and there's somebody who is going to check the papers. They see the papers, they look at the license, they look at the photograph on the license, they look at your face, and that's it. Everything is okay, and you are allowed to take your vehicle in. Here, inside the cell, what is that thing that is checking? Inside the cell, the checks or whatever happens, all the regulations are done by molecules. So, there are molecular checks. In our system, because there is nothing which is like to see something or to ask something. It is molecular check, and especially the checkpoints, cyclic dependent kinases. This is a class of protein which binds with cyclin. Cyclin is the protein which is synthesized when the changes take place. As ATP is synthesized, a little more cyclin will be produced. As raw material is synthesized, a little more cyclin is produced. And as cyclin is formed, this cyclin dependent kinase binds with it. And a complex is formed. This complex is called cyclin dependent kinase cyclin complex. And the amount of this complex will decide whether that checkpoint is open, activated or not. Okay, so this is molecule dependent. Now, let us start with the first checkpoint, G1 checkpoint, which is also known as G1S checkpoint. What is going to be checked? Everything. What happens in G1 phase? We have seen the changes that take place in G1 phase. What was happening in G1? Protein was synthesized, RNA was synthesized, raw material was, everything will be checked. So what is checked? Is cell size sufficient? And cell size would indicate what? Whether all the required materials, raw material, everything is ready or not. Because if raw material and everything is ready, the cell is going to be bigger. So if the size is proper or not, if size is proper, that means all the preparation is done. Are the sufficient nutrients accumulated or not? That means if there is less nutrient, then the cell should not go beyond this phase. Are growth factors sufficiently produced? Is there any damage in the DNA? That also has to be checked. And once all these things are checked, okay, cyclin dependent complex is sufficient quantity, then this checkpoint is activated and the cell comes into the next phase. One very important, if a cell clears G1 checkpoint, then it is committed to divide. This means if G1 checkpoint is cleared or activated, then the cell will divide 100%. You cannot stop that. That means we can stop it before that. So if the cell size is not proper, sufficient raw material is not accumulated, there is less nutrient, then in that case, the cell will be retained in G1 till all those things have been synthesized and accumulated. So this is important. Many times questions have been asked on this also. Now the cell comes into SG. The next checkpoint is G2 checkpoint. And here this checkpoint is going to check for two phases. What has happened in S and what has happened in G2. So here we have written only 2 points, but these 2 points are bigger ones. So it says that we will check the cell size. Again, if the cell size is proper, it indicates whether protein synthesis has taken place or not, ATP synthesis is proper or not, DNA replication is proper or not. Because when the cell divides, both the daughter cells should get same genetic material, complete genetic material. So the replication has to take place properly. So these two things are checked in G2 checkpoint. If this checkpoint is cleared or activated, then the cell comes into MS. And there is one more checkpoint. It is called M checkpoint. It is also known as spindle assembly checkpoint. And the reason is that the checkpoint is basically going to let me draw a chromosome here for you so that we understand that. I'm making a paired chromosome because we want to see the things which are inside it. These are the sister chromatids which we made earlier, and here is this kinetochore. Okay, and inside each sister chromatids there are DNAs. One has the original, other has the copy DNA. Okay. And now the spindle fiber is going to get attached to it. Say the spindle fiber attaches here. These are the spindle fibers. Suppose the spindle fiber is not attached here. In that case, what will happen? One fiber is properly formed, it is going to pull the chromosome towards one side. So this chromosome, which has double DNA, is going to go towards one side, and that will form the cell. So if this is a cell, the chromosome which is coming towards this side has both the DNA, the original and the copy. And the chromosome which is coming here is nothing. There is no chromosome which is coming here. And later on these two will divide to form two cells. So this cell is going to have double DNA and this cell is not going to have that DNA. And DNA means the information. So this cell will also be defective and this cell will also be defective. So what is going to be checked in this point is whether these spindle fibers are attached to the kinetophore or not. Main thing. That is why it is called spindle assembly checkpoint. And what is being checked? Chromosome attachment to the spindle fibers. So if these three checkpoints work properly, then the daughter cells which are formed, they will be without any fault. That means those daughter cells will be normal. And this is what we want because there are so many places in our body where the cells are continuously dividing. And if something goes wrong, then at every place we are going to have defective cells, and defective cells would lead to cancer. So these are checkpoints. What happens in which checkpoint is very important for us to understand. I have few questions for you. Let us see whatever we have studied, all the phases of cell cycle and the checkpoints, whether we are able to answer these few questions or not. This will help us understand whether that concept is clear or not. Okay? In which phase of the cell cycle, in which phase will the cell cycle stop if there is shortage of nutrients? What do you think would be the answer? For the answer, before I give you the answer, let me take you back to the place where we have written this. This is what we were talking about when we were talking about the cell cycle and its phases. All the changes which you saw. We have written everything here, G0. Okay. Do you see something written here? If nutrients are not sufficient, cell is not going to clear the G1 checkpoint. That means you're going to retain the cell into G1. So what is going to be our answer here? The cell will remain in G1. Next question. If in a cell DNA replication fork is formed, which checkpoint is activated? We said our DNA is a double-stranded structure. And when this DNA replicates, it opens and it is seen in the form of a fork. If there is a fork, that means the new DNA is formed or being formed. So if new DNA is being formed, that means the cell has entered S phase. And if it has entered S phase, which checkpoint has been activated? The first one. So we have studied the concept of cell cycle and checkpoints. And using that, now we are able to answer the question. These are interpretation questions. So if you understood the concept, you should be able to answer these type of questions. There is one more. In S phase of cell cycle, what is the situation? Which of the four options you think is correct? The amount of DNA doubles in each cell, the amount of DNA remains same, The chromosome number increases and the amount of DNA decreases. Which out of the four you think has happened in S phase? This is the chromosome in G1 and this is the chromosome in S phase. So there is this original DNA, there is a copy DNA and sister chromatids, original DNA and copy DNA. Has the number of chromosome changed? This was one, this is same one. So number of chromosome cannot increase. There is DNA replication, so there is no chance of decrease in the DNA count. In a cell, if this is the chromosome, there is some DNA content. And if this is the cell and this is the chromosome in the cell, what has happened to the DNA content? It has doubled. So, you have studied DNA replication, you know it takes place in S phase, but when such type of questions are framed, you're using that concept which you have understood to answer these questions. Okay. Sorry. Alright. So let us have a quick recap of what we did in today's lesson. Okay, we started from cell cycle and we said that the cell cycle means the life cycle of a cell. The phases, two, interphase and anaphase. In interphase, there are three, G1, S and G. Before looking at all the written points, we tried to write down the points on our own. And how did we reach to these points? By knowing what is happening in which phase. S phase is very important. In S phase, DNA replication takes place. So G1 phase is the preparatory phase for S. G2 is the preparatory phase for N. So whatever is required in S phase, whatever is required for DNA replication, everything has to be done in G. And what is that? There has to be protein synthesis. Which proteins are we looking at? We are looking at two types or maybe one type of protein in this case, and that is enzyme. Which enzyme? The ones which are required for DNA replication. There are 10 different enzymes which are required for DNA replication. All those enzymes must be prepared. They should be ready for the S phase to take place smoothly. ATP synthesis has to take place because energy requirement is very high. And the raw material for new DNA strand to be formed. Everything done in G1, the cell goes into S phase. All those things are used up for DNA replication. Centriole splits and histone proteins are synthesized. What is the significance of this histone protein? Histone protein is required for packaging of newly formed DNA. That is where we need this histone protein. Everything done, DNA replicated, centriole split, cell comes into G2. In G2, there is preparation for M phase. What is needed? Enzymes are needed. Two main categories. Lytic, to dissociate, dissolve, digest the membranes so that complete space is available for the chromosomes to move. And then synthesizing or synthetic enzymes so that the membrane can be reassembled later on. One more important protein, tubulin protein, which is required for formation of spindle fibers. ATP is very, very important. To take one chromosome from the midpoint to the pole, 30 ATPs are needed. And imagine if we are talking about our cells, then 46 chromosomes have to move from midpoint to one pole, and 46 chromosomes will move from midpoint to the other pole. So 46 into 30 and 46 into 30. These many ATPs would be required only for one division of one cell. And that is why ATP synthesis, ATP accumulation takes. Sufficient amount of ATP has to be accumulated. And then the cell goes into M phase. First the nucleus divides, karyokinases, and then the cytoplasm divides. Two daughter cells are formed. If these daughter cells do not have to divide further, they get into G1, grow, and then go into G0. Then we had all those points written down properly so that if you're making or taking notes while the lecture, you can cross-check whether you have written all the points or not, and you should not be missing out on any points. Then in this S phase, we talked about these sister chromatids, which is again very important. I hope you understood this because this is again a very, very, very important thing. G2 phase, M phase, karyokinases, 4 phases, pro, meta, ana, and tetraphase, and then finally cytoplasm. This particular part we are doing it short because in coming lectures we will be talking about mitotic division, meiotic divisions in detail. So there we will take up all these phases in detail, each and every minor change which takes place. And that's why we are doing it in short because only this much is required when we are talking of the cell cycle. G0, G0 phase or quiescent phase. Again, very important. Many questions are asked on this. Quiescent means inactive. The cell gets inactive only for division. The cell is very, very active only for division. It is inactive. This is important. This cell which we are talking about is very active. It is differentiated cell. It is a specialized cell performing very specific function. But it is not going to divide. But some cells can, like parenchyma. So when differentiated cells come back into the cycle, we use a term dedifferentiation. This is again something which we will be discussing in coming chapters. Duration can vary depending upon the type of the cell that we are talking of. There are 3 checkpoints to assure that proper division takes place. And the point which we made here, the cell is committed to divide if it has crossed G1S checkpoint. Very important. All checkpoints are designed in such a manner that nothing wrong takes place. Everything is properly done and the new cells which are formed are proper. And then we had some practice questions. I gave you 3 questions. If you were not able to do or solve these questions properly, I would suggest that you revise the basic concept of cell cycle and checkpoint and then try to attempt these questions. Okay? So, this is what we had for today's lecture. I hope these 2 concepts are absolutely clear to you. Now, if you plan to subscribe to whichever plan you're looking for, monthly or 3 months or 1 year, don't forget to use this code so that you are able to avail that 10% discount. Okay? So, in the next lecture, we will continue with the division part, and till then, keep revising. Don't forget to draw the diagrams. I'll see you in the next class. Till then, take care and keep revising.